The overall goal of this procedure is to prepare Drosophila melanogaster embryos for whole mount visualization of the nervous system. This is accomplished by first collecting embryos from the fly strain of interest. The second step of the procedure is to remove the corium with bleach. The third step of the procedure is to apply heptane and methanol to remove the vitelline membrane. The final step of the procedure is to apply antibodies to the embryos. Ultimately, results can be obtained that show the structure of the Drosophila embryo nervous system through immunofluorescence microscopy.
This method can help answer key questions in the field of neuroscience, such as what role mutations can play in the developing nervous system. Generally, individuals new to this method will struggle because they may accidentally pipe out embryos from the vial.
Begin this protocol by preparing the reagents that will be needed, including PEMFA buffer, PBT, 5% BSA with 0.01% sodium azide, saline solution, and formaldehyde fixative. Prepare a cage for the Drosophila strain of interest by knocking the flies out with carbon dioxide, and then transferring at least 100 flies to a plastic bottle containing an adapter with an apple juice agar feeding plate. Place the cage of flies in the dark at room temperature for 72 hours. Change the apple juice agar feeding plates every 8 to 12 hours, so that the flies get acclimated to the cage and do not hold their eggs. For the experiment, after the 72 hours